I think we can go ahead and get started.
All right, yes, thank you, Hannah. So welcome everyone to the Data Core webinar on innovative neuroimaging methods in AD research. We have three speakers that have planned like amazing talks. We'll start off with Chris Schwarz from the Mayo Clinic, followed by Kiran from Wake Forest, and Hui Zhang from the Northwestern ADRCs. And you'll hear a wide range of neuroimaging methods research in their talks. So without further ado, Chris, floor is all yours.
All right, hello, thanks for coming. I'm going to talk about potential re-identification of brain imaging research participants using face recognition, so risks and how to mitigate them. In short, the defacing talk. Public sharing of research data, including imaging, is being widely promoted and sometimes required, as most of you well know. Typical de-identification of brain MRI removes only text metadata stored in the image file. So you have a DICOM, that DICOM would have the patient's name, the scan date, age, sex, study site, all kinds of stuff. And old style de-identification would just change that to a participant ID. In many cases, you'd leave the date alone. Sometimes you'd get rid of the scan date. The other stuff usually stays. But that would be considered de-identified. That's the end. The image would be left entirely intact. And our research question was, is this de-identified enough? Could somebody re-identify the person using the imaging data? HIPAA standard for de-identification requires removing, quote, full face photographs and any comparable images, but they don't really define what is a comparable image. So our research question was, is an MRI or a brain MRI specifically now going to count as a comparable image? So if we think about a research study, something like ADNI, but many others as well, that have de-identified health information is released by a research study. It's including brain scans, but it's also got biomarkers, blood panels, cognitive testing, diagnoses, family history, maybe your complete sequence genome. And here in the modern era, most people have public photographs and identities off on the internet. Just get it off of Facebook or what have you. If you suspected that somebody was in that data set, could you take all of the MRIs in something like ADNI, create a 3D surface reconstruction out of each one, and match it with photographs that you might already have of this person in order to find them in ADNI? And if you did, then their public name would be reassociated with that de-identified PHI by matching photographs with face recognition. Why would anybody want to do this? Well, sometimes it's really innocent stuff. Like, say your dad is in this study and you're getting a little worried about him. He's not thinking as well as he used to, and you might be tempted to find out his amyloid status. Maybe you yourself are in the trial, and some clinical trials are putting data out for data sharing before they're done. And you yourself might be tempted to pose as a researcher and see if you're getting the placebo. More invasive stuff. Employers would love to know who to fire by who might have a risk of Alzheimer's in a few years. Corporations love to sell targeted advertising. Or the more far-fetched stuff, say Joe Biden needs to enroll in a trial. I suspect Vladimir Putin would like to know that. But we want to make sure that everyone feels comfortable to enroll in clinical trials and in observational studies without worrying about their data being re-identified for any of these kinds of preventable reasons. So we took a typical face recognition problem where you would provide examples of a training set of photographs of faces to be recognized. Each would be labeled. You'd have a few examples of each one. You'd give it a test photograph of an unknown individual and say, computer, match it to the correct face. And the unknown person becomes George Bush. We flipped this on its head and said we provided examples of a training set of MRI-based face reconstructions of faces to be recognized, each with participant IDs. And then we give it a test photograph of a named individual. We'll pretend his name is John Smith, and we'll say, which one is John Smith? Oh, great, it's participant 52352. We'll count up how many times that type of query got the correct answer. thereby re-identifying that person in a data set. Our original data set, we had 84 people from the Mayo study of aging between ages 34 and 89. Each had a previous head MRI within 3 months as part of their existing enrollment in the Mayo study of aging, or ADRC. And these were Siemens 3D FLAIR scans. They were identical to ADNI3 protocols. These were not special face optimized kind of things. These are 3D FLAIRs, like any normal thing you would do for brain research. We captured 5 photographs of each volunteer with a standard iPad. We just had them look up, down, left, right, and center. Because if you were trying to find a person in a data set, you could probably get more than 1 photograph of them. So MRI-based face reconstructions, we just have a handful of examples here of photographs, eyes open, eyes closed, and what do their corresponding MRI look like. MRI largely preserves shapes and relative sizes of facial features. It does not have hair, lighting, or skin pigmentation. These are things that humans, when you look at each other and try to recognize a face, you probably use features of their skin or their hair. Those things are not preserved in an MRI. And there are some shape deformations due to supine position. You're laying down, everything slides back. Or touching of ear padding and head coils, because you might be squished like this, or you might be squished like this, and those things come out in the MRI. But computers, automatic face recognition, aren't necessarily using these kinds of features that you, a human, might use. Some humans look at these images and say, oh yeah, it's obvious which one is which. Other humans, not so much. But the important question is, could a computer do it automatically? So we generated 81 2D photograph-like images of each MRI using SurfIce and under varying simulated lighting positions and views. And we use this as the training set of faces to be recognized. We tested using software that, at least at the time, though maybe not anymore, was available easily and freely to the general public. We used Microsoft Azure face recognition. At the time, it was a free demo, as long as you didn't do more than 30,000 faces a month. But we did this within a private, secure Mayo Clinic cloud instance to protect the data, kept all the PHI at home. And the results were that for 70 out of 84 participants, that was 83%, their correct MRI, which was the FLAIR, was chosen as the software's #1 match for their 5 photographs. Matching by random chance would expect only 1 correct match. So that was a really nice p-value, and that was our original study published in New England back in 2019. Since then, we have replicated that with 182 people. We also added PET and CT. We updated to Microsoft's even newer face recognition, because even just on the scale of a couple of years, this stuff gets better and better. And the match rates for FLAIRs and T1s are up to 98 and 97%. Again, we've got some examples here. You can judge for yourself, but the important question is how well the computer did, really. 98, 97% on data sets of 182. We also looked at PET. So these quote unquote older PET scans that we've got an example of here are about ADNI3 generation. They kind of cut off below the nose, 8mm kind of point spread functions. Amyloid PET, PIB, you could do it about 32% of the time and get the right answer. For Tausipir, you got about 35%. FDG, you got about 41%. Not too significantly different, but frankly, a lot higher than we had ever thought it was going to be at the time. Grant reviewers said I needed to look at PET, and I was like, no way. But I was wrong, and they were right, because we could do it with PET. And when we look at even newer PET scanners, we've got a Vision 600 from Siemens with this kind of PET MR incorporated technology, even though it was a PET CT scanner. And these newer images are really starting to look much closer to MRIs too. And even though our sample sizes are very small, it's 17 out of 20, 14 out of 14, 18 out of 19. This was a problem with the older generation scanners, and it's going to be even more of a problem with the newer generation PET scanners. CT is not a surprise to anybody. People have been doing this research in CT for much longer than me, but we got about 78% with these CT scans that were from the attenuation correction scan of a PET CT. If you had a clinical head CT, it would probably be quite a bit higher. We tested on a variety of MRI sequences, because something like ADNI says, great, which MRIs do I have to deface? Well, that's definitely all the 3D sequences, be that a T1, T2, or T2 flare, are looking at 98, 97, 96%. When you get down to the older 2D sequences, your 2D flare, your or even a MEGRE, you're looking at about 44, 45%. If you got about half a face, you probably got about 40%. T2 stars, about 10%, really just not as good a contrast on a face for a T2 star. And some good news for the folks doing functional MRI, ASL, perfusion, or diffusion. Really almost no risk of face recognition with those. I've tried really hard to get a face out of them, and at least with current generation sequences, they are not a risk. Maybe someday, I can't speak forever, but for right now, for the things that are floating around in ADNI3 and ADNI4, those sequences are not a problem. Okay, so what do we want to do about it? Defacing, as you might guess. Software to remove identifiable facial features in MRI, aka defacing, have been available since about the mid-2000s, but mostly were not applied in studies of aging in the US because a few reasons. People thought that re-identification would have a low success rate or nobody would do it. Researchers were concerned that removing the face would reduce the scientific utility of the data. And the existing software kind of had minimal validation. Nobody really tested whether it would prevent face recognition. It was thought that if you removed at least most of the face and you didn't touch anything in brain, it'd be good enough and it wouldn't harm any of the brain measurements. Most of that turns out to not be true, and that's what I'll show you. Ideally, defacing software protects participants from re-identification, and we want it to not degrade any measurements of useful data. So we want to run FreeSurfer on the brain scans and get the exact same answer. People have known for a long time how to use an atlas to identify face voxels. It's not that hard. But how exactly do we alter them to prevent face recognition? This is a few examples of other defacers and what they've removed. There is one called mask face that was used in some HCP data that blurred only the outer face contour. And some work before mine was able to show that you could automatically unblur that to recover the original face with a convolutional neural net. This stuff's trickier than people ever gave it credit for, is really my point here. So our solution that we came up with, we called MRI reface. We replaced the face with an average face rather than remove it in an attempt to produce a natural looking image, because we thought that would reduce the effects on pipelines downstream. Something like SPM or FreeSurfer or FSL was never trained with images where the face was chopped off. They were trained with images of a whole head. So if we give it a whole head with a whole face, maybe we'll get more of the right answer. We use nonlinear registration. We can more precisely find face voxels. And we also try to replace ears. Ear recognition is a thing. Teeth, any dental record kind of stuff. Aliased wrapped face parts that might be floating around in front or behind the head because it's MR and that stuff happens. And we supported T1, T2, T2 star flare, FDG, amyloid, tau PET, and CT, unlike all the other defacers that are really just doing T1 and anything else is a happy accident. We support DICOM or NIFTY, and we've got binaries or we've got a Docker image, making things nice and easy. But really our point was that the replacement face tries to match the properties of the original input image, match the contrast, match the bias fields, and match the noise. This is free on Nitric at MRI reface. Just Google that and you will find it. So the validation, we repeated our face recognition testing with deface data using our MRI reface, comparing it with MRI deface, which was FreeSurfer's method, FSL deface, and Pi deface. MRI deface and Pi deface mostly prevent face detection, but they often retained the eyes, etc. It would just remove the nose and everything below it. In order to test whether that would be good enough, we warped a template to the defaced images and we filled in the missing face parts from the template so we could test face recognition from the remaining partial faces, see how well you could still re-identify people after these partial defacings. What we found was higher than even we expected, but these other defacers, we could do between 28 and 38%. We could still re-identify after the defacing. Ours on our original version was a similar 28%, but since their original publications, that's come down considerably, down to about 7% as we replaced more of the eyebrows and forehead. 7% still exceeds chance, which would be 1%, but it's a heck of a lot better than 97%, and we are continuing to improve this all the time. We ran each defacer on 300 ADNI T1s, 100 from each vendor, 50 controls, 50 clinical ADs. We ran SPM12, FSL, and FreeSurfer 6 on each original and defaced image. And we compared the regional gray matter volumes before and after. We looked at the interclass correlation coefficients and compared them against a couple different notions of scan-rescan. These yellow boxes are the back-to-back scans from ADNI 1. They were 3Ts, back-to-back 3T T1s. And all the defacers came out far better than scan-rescan, which is good news, but we're the pink box on the right, and we were smaller than the other defacing software for at least 3 out of 4 of these comparisons.
Sorry, time check. It's 3.
Okay. All these effects are larger than you would think, given that nothing is actually messing with the brain at all. Defacing affects brain measurements because it affects the affine registration of the template, which is affected by the face, especially the nose. The eyebrow ridge is the most important part to face recognition. It's also within a few millimeters of the frontal lobe. And we do see the largest effects of defacing are in the orbitofrontal and the frontal pole. Also, Bayesian tissue class segmentations like those in SPM compare everything in the image to everything else, because in MR, everything is, all intensities are relative. So if you change anything in the not gray matter intensity distribution, it will affect the probability of being gray matter. And this causes the largest effects in regions that are the most difficult to segment normally, like the deep gray and the sensory motor. Our latest versions include support for CT and PET. We reduced PET recognition rates down from about 32 to 41%, down to 0 to 3%. CT gets down to 5%. And as we said, MRI gets from 98 to 8%. The effects of defacing on a, say, a global PIB measure are far, far below scan-rescan and pretty much negligible. We suggest using our software for all of these image types that are necessary. And I will stop. Thank you.
Yeah, thank you. That was amazing. Very clever technique of using an average face to, get rid of that. We have a couple of questions. One is from Dave Nguyen. He asks, are your replacement faces sex and race specific? Because if they are, then it may affect racial bias in the models based on these MRIs.
Replacement faces were derived from the Mayo study of aging. It is largely a Northern European population. We are aware of that limitation, and that is one of the aims of our grant that we've got to try to fix this. Some of the ideas are to be sex and age specific replacements. On the other hand, other ideas are to try age and sex replacements that are deliberately not the same age and sex, because this can make it harder to recognize people. These are somewhat competing aims between preventing re-identification and trying to have things best resemble the original to reduce effects on FreeSurfer and the like. But we are testing all kinds of things, and stay tuned because that is our biggest focus right now.
Great. And another question is the comparison to chance here really reasonable? You have a close small group, and which is not the real world where your possible patients could be more, much larger. So can you use this method to identify anyone that was not in ADNI, like outside your training set? So in the interest of time, maybe you can answer that in the Q&A, Chris, and we can move on to our next talk, and that would be Kiran from Wake Forest. Yes, thank you. That was amazing.
I think, Chris, you need to stop sharing for me today.
Got it. There you go.
Thank you so much. Go ahead.
Right. Sorry, I have multiple screens. All right. Thank you, Sudeshna. Thank you, Hannah, for the invitation. I'm talking about my project here and my research exploring microtubule-based PET for Alzheimer's disease imaging. So I'm going to give you a brief outline of all the work we have done here, all the way from scratch, literally from the bench to the bed. So we have designed and developed a tracer from scratch and then moved it to validation from rodents to monkeys and then to humans. So again, we all know the neuropathological hallmarks of Alzheimer's disease are the amyloid plaques and the tau. And again, these are the two common neurobiomarkers for PET imaging. So we still use them regularly for many of our disease for the AD pathogenesis. However, tracking AD pathogenesis in a clinical setting based solely on the amyloid plaques and tau is not greatly appreciable, meaning it's challenging, especially because of the uncharacteristic regions of the AD pathology lying in the early or mild cases, that is the MCI and the early AD cases. So especially we are talking here in the preclinical AD, where you can see that the pathological cascade of AD begins almost 15 to 20 years before the clinical symptoms appear. So it's very important to really understand the early diagnosis, that is understanding the pathogenesis of AD much before the clinical symptoms even come up. So in that quest, in that search, so what we have found is maybe microtubules might be a wonderful target. What are microtubules? Microtubules are these cytoskeleton polymers comprising of repeating non-covalent bound alpha and beta subunits. So they're very important in a multiple functions of cellular trafficking, signal processing. As you can see in this particular figure, in the healthy microtubule, the complete microtubule filament is well protected and in a particular shape is well maintained, whereas in Alzheimer's, the microtubule is completely disintegrated and you can see there is no more tau protecting it, and you can see hyperphosphorylation happening. And the disruption of microtubules are implicated in multiple neurodegenerative diseases. I'm not going to read all the names, but predominantly AD, PD, CVD, all these diseases have implications of disintegrated microtubules. So therefore, the lack of microtubule binding imaging agent hampers the drug development for brain diseases involving especially the changes of microtubules. So what was the main thing? If you really want to see the microtubules, you really have to find out a radio tracer that can go in the brain and quantify microtubules non-invasively. So again, we were not the first one to develop a microtubule radio tracer. It's already been there. Paclitaxel, docetaxel, again, these are the microtubule agents commonly used for many cancer diseases, treatment of cancer diseases. But what was the problem? So none of these tracers were going in the brain. Very poor or no uptake in the brain. So almost limiting their utility in the CNS imaging. So therefore, our idea was to really develop a radio tracer, which is brain penetrant. So on that quest, we found this particular tracer, this particular compound, MPC6827, a microtubule targeting agent used for several cancers. And again, high affinity, it has very high microtubule affinity, as you can see, with 1.4 nanomolar affinity, ideal pharmacokinetics, and most importantly, a clinical information are already available. It means it is a drug in phase 3 clinical trial of GBM, and again, removed because of its toxicity associated with some other cardiovascular diseases, cardiovascular dysregulations. But most importantly, as a radiochemist, I look at the structure and see it has an amenable methoxy group here, where you could easily label with a C11 radioisotope. So we went ahead and made the precursor and radio labeled the compound with high specific activity and a high radiochemical yields, very reproducible synthesis, which is where we can move forward for further translation studies. So then moving to the next part of our translation, we made the compound. Now we go to the validation stage, that is in rodents. So as you can see here very clearly, in the first, we did it in the normal mice, clearly goes in the brain in the particular baseline studies. And the next, we even blocked with the cold compounds of the first generation and second generation. More than 80% of the tracer was blocked, showing again our tracer is very specific to what our purpose of microtubules. And again, we did the same thing in mice and rats. Both of them shows clear uptake in the brain. This is the first blood-brain barrier penetrating PET radioligand for imaging microtubules. So then again, this has to be corroborated with ex vivo studies. We did the pharmacokinetics where we did like total body parts and got their gamma count, got their counts using a gamma counter. And you can see an ideal pharmacokinetic ratio where from 5 minutes to 60 minutes, it really washed out very well. And again, the same blocking study was demonstrated using a ex vivo study, clearly showing well compatible ex vivo studies when compared to the in vivo studies as well. So then with this utility of microtubule MPC, we want to see what happens in AD mice. So then we made sure we got the tracer into the brain, and now we want to see what exactly is it binding. So we did the mechanistic actions where we took the cells, the neuroblastoma cells of SH-S5Y5, the neuronal cells. We did some microtubule upregulating and downregulating, meaning the stabilizing and destabilizing agents. With the destabilizing agents, the uptake increased, whereas the stabilizing agents, the uptake decreased. It almost shows that this particular tracer likes in an environment where there is more destabilized microtubules. So maybe C11 micro MPC 6827 measures the microtubule destabilization. And again, the same in vitro mechanism was corroborated with the in vivo micropet imaging, where the same animals at baseline showed a decreased uptake when we treated the animals with ApoD. ApoD is a microtubule stabilizing agent that stabilizes the microtubules, epithelium D. So this is an interesting study where we did the longitudinal study from almost 4 months to 15 months in APPPS1, which is an A-beta overexpressing mice, and P301S, which is a tau overexpressing mice. In both the cases, we have seen that from 4 to 5 months till the increase to 13 to 15 months, in the transgenic mice, the uptake increases. The same mouse at the earlier time points and the same mouse at the later time point shows an increase in uptake. So more noticeably, a few things I do want to bring it up is in both the cases, you see an uptake, which is showing that microtubule disintegration drives both A-beta and tau-induced neuronal dysfunction. Because this is APP is only amyloid beta and P301 is a tau overexpressing mice. And more importantly, if you come into the P301s, so you really see a changes in phosphotopathology from 7 months, whereas this particular tracer could see the differences within 3 to 4 months. Again, showing that the microtubule destabilization may precede the phosphoto formation in AD brains. So this is again supported well with the tau knockout mice we did, where tau knockouts have higher destabilized tubulin expression, and we have seen the same thing. So again, corroborating what we saw in prior discussions that MPC 6827 measures MT destabilization. So then because we want to move it further, we had to find the translational aspects of binding constants. Both KD and Bmax were well favorable parameters, were in the range of acceptable parameters for a CNS-based radio tracer. This was done all in the rodent brain tissues. And then we also did some metabolite analysis showing that the compound is almost stable with 98% in rat plasma, and more than 96% was stable in rat brain, meaning like what you see in the image is the intact compound of MPC 6827. So the story is good until the rats. Now we want to see how does it behave in the monkeys. So we did first in some images in the common healthy 4 male vervets, and we saw that the uptake was very much favorable, as you can see here. The high brain uptake and favorable pharmacokinetic properties, including the peak activity within 5 minutes, like you can see. And more importantly, very much like the test and retest scans were reproducible. So having this in hand, we went with the PET imaging of non-human primates model of AD. That is, as we took 4 animals, which has different CSF A-beta numbers, the lower A-beta numbers and the higher A-beta numbers, and again, with different ages. Vervets with the low CSF A-beta, that is presumed higher plaque burden, showed higher radioactive uptake, as you can see here. Whereas compared to vervets with a higher CSF A-beta, again, presumed low plaque burden, showed much higher uptake here. So we showed that the youngest vervet has the lowest uptake, and MPC 6827, the uptake was inversely related to the CSF A-beta values and directly related to the age. So most importantly, what we did was the same animals, and we got the brains out, and we did an autoradiography studies, also corroborated what we found. That is, more uptake in the AD-like monkeys than in the control monkeys, which has more CSF A-beta values. So now our hypothesis is MT destabilization might be happening prior to amyloid beta plaques and tau tangle formation. So maybe MT-based changes are linked to both hallmark pathogenesis of tau and plaque deposition. So on that, we went also another important application, I do want to bring it up, is a lot of MT stabilizing cancer drugs have been repurposed to be used for preventing axonopathic changes. Like for example, ApoD is being used in clinical trials to reduce some axonal transport deficits and has shown some improved cognition. And also another compound TPI 287, a microtubule stabilizing agent, reduced amyloid beta deposits and neuronal loss. So it almost makes it clear that the normalizing pathologically disturbed microtubules are very important as a treatment strategy. For another application of imaging microtubules is PET imaging could track the real-time destabilization, and it could be an optimal AD therapeutic development. So with this, we move to humans. As before we did into AD humans, I want to show something we did with the normal healthy humans we started with. We want to get the dosimetry study done. This was done in Columbia University with our collaborator, Dr. Akiva Mins, where we did PET imaging in 6 males and 6 females, and it showed up a very acceptable dosimetry numbers, not just in brain, throughout the whole body, which we do, like at least 13 organs have been measured, and standard parameters are well in the acceptable range for a CNS radiotracer. So having this data, we went ahead and did some cognitively normal and impaired older adults, just as a pilot data here at Wake Forest. So we first selected 2 cognitively normal subjects, which has no impairment of no impairment on the memory and with objective testing especially, and they have the A-beta PET values SUVR of less than 1.2, meaning they are cognitively normal. And the cognitively impaired one, the MCI, the MMSE score was greater than 21, and CSR and the A-beta PET was greater than 1.21. This was our criteria, and we selected 2 from each category to see whether the proof of concept, it works in those subjects as well. As you can see, when compared to cognitively normal, both the MCI subjects had a higher uptake. So not just in whole brain, we have seen that some parts of putamen prefrontal cortex and frontal cortex and hippocampus showed more uptake when compared to cognitively normal. So again, we need to make sure that our new radio tracer is well aligned with the existing biomarkers. We then did an uptake correlation with the age and beta 181 and C11 PEB and FTP SUVR. As you can see here, it is directly proportional to the age and the A-beta PEB value and the tau PET values, and inversely proportional to the CSF A-beta. Again, well relevance to the AD biomarkers. And most importantly, we did a scattered plot, which we show that the MPC 6827, that is the MT destabilization, happens much earlier before you see an elevation in the FTP here. Before the elevation here, you can see that the MT destabilization has already happened. So again, showing that MT destabilization might be happening before the FTP uptake is taking place. So again, this is well corroborated with the human brain autoradiography as well. We have seen that both in cognitively in MCI subjects in prefrontal cortex and hippocampus, there is slightly higher uptake when compared to the cognitively normal subjects. So uptake increased with MT destabilization, which is what we have seen in both rodents and monkey models, and radioactive uptake was inversely related to CSF A-beta levels and directly related to age, may precede phospho-tau formation, and we are currently collecting data to do more subjects to exploit the clinical significance. So apart from this, we were also doing, I'll just run with this within 2 minutes. I know we are running out of time. So this, we are currently exploiting our microtubule imaging with another important marker of synaptic dysregulation. This is what I presented at the AIIC, where we see that the 2 important biomarkers of SV2A and microtubules are considered here for, again, as an early biomarker, and we use them as our changes. So dual PET imaging has been done. What we have done here is our experimental model is very simple. We took the same animal, that is the APPPS1, which is an amyloid beta overexpressing mice, And in the morning, we scanned the animal with MPC6827. In the afternoon, we scanned the same animal with UCBH. We did at 4 to 6 months, and also we repeated the scans at 9 to 10 months. And corroborate with this, we also did some behavioral testings. To our surprise, not surprise, we found that the MPC6827, the transgenic mice has a higher uptake when compared to the wild type one, as you can see here. And it did the exactly opposite when it came to SV2A. That is, the transgenic mice had a lower uptake of SV2A than the wild type mice, which are again age matched well. So not just the imaging, we also showed with the biodistribution and autoradiography, same thing was corroborated. That is, a transgenic mice had a higher MPC uptake, that is a microtubule disintegration, and SV2A was lower in the transgenic mice. So we're still doing the work in progress. And more importantly, I do want to bring it up that our microtubule and synaptic radiotracer can bring up changes much earlier, whereas the behavioral changes, as you can see with the nesting experiment, can only be seen at 9 to 11 months. So maybe imaging strategy could potentially track the early AD pathological changes, both the microtubule destabilization and SV2A levels. And this is what complemented is the same animal want to show that higher MTD stabilization and lower SV2A levels on the same day. Another important strategy is this particular thing can be used for a drug treatment strategy. That is, can a single drug molecule target both? Like, for example, microtubule stabilizers improve synapses, or can A-beta tau targeting drugs improve both? So that's work in progress right now. We are currently pursuing this option where we are doing the AD mouse model, we are doing the baseline and treating the animals with a lot of with this particular drug of MT stabilizing and synaptic drugs, and we're also doing some behavioral tests and want to see whether it comes back to normalcy. Before I end, this is all the acknowledgements. It wouldn't happen without my group, and these are my group people: Naresh, Bhuvan, Avinash, and Krishna, Ivan, and Mac, and Translation Imaging Program for helping me with all the image acquisitions. And my collaborators and two key collaborators are Dr. Kraft and Dr. Mintz, without whom this wouldn't have happened. A lot of people to be thanking, and my funding sources. Mayo one for getting me some data on my mouse data and my pilot grants. Thank you.
Thank you, Dr. Solingapuram. That was fascinating. So microtubule-based PET imaging, is that like really getting used a lot for like early detection and clinical trials, or you're in the leading edge and we're yet to see?
We are yet to see. I think we are currently exploring some mechanisms to get some funding to see the larger population. We just did very early. I think the very early data is very promising, and want to see if we can extend with more subject. Not even like a clinical trial. First, we want to see the imaging study in a larger cohort and see how good it is. What we see in 2 subjects can be reproduced in more subjects.
All right. Okay. No, that's great. So we do have a question on the Q&A platform, but let's move to Professor Zhang. So if you can share your slides, and if you could answer the question on the platform. It's a question that I had as well, is why is the uptake higher in the disintegrating microtubules as compared to the healthy microtubules? So feel free to answer it in the Q&A. Thank you. Please go ahead. I think I see a little bit of your slides getting cropped to the left. Is it just me? Hannah, do you see that same issue?
Oh, I see. Maybe I move this.
I think you weren't in full screen. It's cropping it into a square.
Oh, I see. I think most should be fine, and I move a little bit. So good morning or good afternoon, depending on where you are. And thank you for this come to this webinar. And my name is Hui Zhang. I'm a professor of the biostatistics at the Northwestern University. And I work with this Alzheimer Disease Center here at Northwestern as the director of the core for the data management and statistics. Today, I'm going to talk about the unbiased and robust analysis of correlation in Alzheimer's disease research images. So if I see more top, I will let you know. Okay, so for the Alzheimer's disease, so when the very important protein is called the amyloid beta. So amyloid beta and has lots of interaction with another protein called TDP-43, which regulates the expression of amyloid beta. So their interaction is a very important research topic for the Alzheimer's disease. For example, this paper published on Nature Communications a couple of years ago. So in this paper, for one of this image, the figure 8, they show the amyloid beta at green color here, the TDP-43 with the red color here. And to see their interaction usually indicated by they combine with each other, or in another term called the co-localized. So how to show they are co-localized? Because when the green color and the red color merge together, they show yellow color. So when they see the yellow color, we know, oh, okay, here's the interaction between the amyloid beta and the TDP-43. So I guess probably we have 130 audience here. Probably not everybody know the biology very well. So we have two questions. So now, how do you label your amyloid beta with the green color? How do you label your TDP-43 with the red color? And when they are together, why they show the yellow color? So that's a perfect question if you are not very familiar to the microscope and the biology. So I want to explain this. So for this image, this is usually from confocal microscope. So confocal microscope use the laser. So laser usually is a light with a very short range of wavelength. So that's why I want to use one slide to review what we learned about the light and the wavelength. This is probably everybody learned in the high school physics. So the light is actually a wave. So for the visible light, it has wavelength probably from 400 nanometer till 750 nanometer. So for the frequency or the wavelength will determine the color of the light. That's how we could see. So light is a wave. In addition, light is also particle. That's what we learned in the high school physics. So as a particle, the light has energy. So the relationship between the energy and the wavelength is described by this. So this is H and C, the constant. So the energy is reversed to the wavelength. Okay, so how do we label the amyloid beta with green and also label TDP-43 with red? So that's we use some fluorescent protein. That's actually Nobel Prize in Chemistry in 2008. Because we found the biologists found this protein. We use the laser with a short wavelength. Short wavelength means high energy, as we mentioned, like a blue color. And this protein absorbs a little bit of energy and releases less energy. So the less energy is less than this energy. So it will have a longer wavelength. For example, they can release green color. So that's why we can see this protein as a green color. By some biomedical engineering, we attach this green fluorescent protein to our target protein, like amyloid beta. That's why we can see the amyloid beta under the microscope. And the biologists are very smart. They revise this protein a little bit and get also the red fluorescent protein. It will emit red fluorescence. So that's why how we label the amyloid beta and also TDP-43 with green and red under the microscope. So again, let's return to this. If one color is green, one color is red, when they merge together, they will go to the color in the middle, that's which is yellow color. That's why we see the yellow color means they are merged together. These two proteins interact with each other. So that's a confocal microscope, use the laser light. So for the large microscope, actually the best resolution they can reach is 250 nanometer. You cannot get lower than this, the resolution, because light has a wave. The wave has some physical feature called the diffraction. That's why the limitation. That's a limitation for the confocal microscope. So how to further improve the resolution of this microscope? That's a Nobel Prize in Chemistry in 2014. They use some algorithms, use some techniques. For example, this is normal distribution. They cut it off to tail, they only use the middle one. They further improve the resolution to 20 to 30 nanometers. For example, that's a confocal between this improvement. This you can see the blurred microtube or whatever. Dr. said that mentioned microtube. And after the resolution improvement, you can see the microtube much better, much clearer. So that's a resolution improvement. But after this, for the confocal to show the correlation between red and green protein, we got the trouble. See, previously, if with the standard confocal with resolution 250 nanometer, If you see the red or green, they merge together through the yellow, you see, okay, they are colorized, they are interactions. That's called a colorization. And then they give you an answer, okay, yes or no, will this green protein, yes, red protein together or not. But after this high resolution, the signal is much clearer. So that even they are very close to each other, they won't be the color won't merge together. They won't show the yellow color anymore. In addition, here the yellow color I'm going to give you a binary answer, yes or no, for the colorization. But sometimes we want to know the answer is quantified answer. For example, Pearson correlation, 0 means that these two variables have no correlation. Like a 0.9 would mean the very strong correlation. A negative 0.5 means negative correlation on different direction. Public polarization, sometimes they have no relationship. Sometimes they try to be polarized very tightly or loose tightly, less tightly. Or sometimes they try to repel from each other, not random distributed. So could we quantify this polarization use some index, like from -1 to 1, like a Pearson correlation? So we checked the literature, we didn't find any statistical method available. That's why we try to develop statistical method for this to address this question, help the Alzheimer's disease research, because I'm the professor of the biostatistic. So that's why we published this paper last year on the journal called the Statistical Method in Medical Research. 2022, we published this paper. So if you are interested, you can take a screenshot for this one and find this paper. That's why I won't go through all the detail, mathematical, technical detail for this paper, because I guess most audience probably they don't have a mathematical background. But let me introduce our method a little bit. So this is with a high resolution microscope, this is what we can get under the micro. This is a real data image. One protein labeled with red, another labeled with green. Because resolution is so high, we can get the location of the single protein molecules with a 2D or 3D coordinates, XY or XYZ. So with these coordinates, actually we can reconstruct this image, use the mathematics, get this image. So by this image, so now we want to quantify the polarization between the red and the green protein. So how to do that? So the very straightforward method or thinking idea is, okay, we use something like similar like a Pearson correlation. If we find the correlation between the red signal and the green signal, if correlation is like maybe 0.9 means they are very strong polarization, negative 0.5 means that okay, they repel from each other. However, we will get some trouble. For example, if we simulate data, use the computer generate some completely special randomly distributed signal like a red or green. So when the density is very high, this Pearson correlation always go to 1, close to 1. But that's not true, right? The Pearson correlation should be 0 because both signals are completely spatial randomness. So that's why this method won't work. Another challenge using this method is that some signal is on the edge of your microscope field, on the boundary, close to the boundary. For example, for the red one, so for this red signal, you won't have lots of chance, you won't have any chance to see the green signal here because it's a boundary, right? So this we have a neighborhood or kind of boundary effect. So because it's too challenging, the Pearson correlation won't work. That's why we propose our method to address this, the edge effect correction, and also for the density correction. This is some technical details I want to go through because based on the limitation of time. If you are interested, you can read our paper. So after we got this index, we want to normalize it from -1 to 1. So in that case, everybody will see this index, they can immediately tell, okay, how strong is polarization or repel from each other. That's why we did some normalization. Why we use this value minus the mean and standard divided by standard deviation. This get a Gaussian distribution. That's called a Z-type normalization. Or we do the ratio get the normalization. That's why, based on this development, this is the mathematical improvement that we proved this, we developed this, we defined a new index called the normalized spatial intensity correlation index. We call the NACC. So for the Z-type normalization, this is how do we define it? Mathematical formula looks like a complex. If you are interested, you can read our paper. This is for Z-type correlation. For the R-type correlation, the definition is here. Or we call the D-type correlation for the ratio. So now, the very important question is become, okay, when you see the red and green signal, they are co-localized. You need to give a definition polarization. For example, how far would be the green? From red, you will claim polarization. For example, for this image, if the red and green is within the neighborhood of like a 1 millimeter or 1 million nanometer, you claim polarization. Oh, everybody here will perfectly correct. But if you claim the neighborhood range is like a 1 nanometer, for nobody will correct. So to determine the size of the neighborhood is very important. In this paper, we give different suggestions, 3 different suggestions how to define the neighborhood to claim polarization. So what's this? We want to compare some other method. We found before our paper, the best method is called the MCDC method. They can study the correlation between 2 signals in a 2 dimension. So this is the best method we can find before our paper. So we want to compare our method with this paper. So we perform a simulation. Simulation is the regular technique we use for the statistical method. We use computer to randomly generate data, and then we test our method performance. For example, we generate data for 4 different scenarios. This scenario, the red and green signal are completely spatial randomness. They have no relationship at all. For this one, the red and green, they have a very good overlap, the 2 clusters. And this is very good polarization. And this one, they are not randomly, they are trying to be away from each other. And this one is the partial overlap. This is the very most difficult case. And we compare our method with the other method. You can see these 4 are our method with different neighborhood definition. These 3 are the other method. So for the first one, of course, all the methods almost give you a very strong correlation. That's reasonable. This one should be spatial complete randomness should be 0. Our method give you the value very close to 0, but some other method mess up here. But the most important for the exclusive one, for the exclusive one, it should be negative index. Our method give you a very negative index, but some other method give you a very positive index, which is completely wrong. So this is our simulation. And this is for one simulation scenario. The other simulation is like this. It's more difficult. If you have the blue signal, green signal, at the center they are polarized, but on the 4 arms, they are trying to repel from each other. So this is much more difficult. We also compare our method with other methods. And also sometimes maybe we have more than 2 signals. We have 3 different signals, like a blue, red, and green. And our method can still work, but no other method can work for these 3 signals. So this is basically our method. We use simulation, we demonstrate that it works very well and beat the previous method. And I know not everybody is good on coding. Our index definition very complex on mathematics. That's why we provide the R package. If you know R, you put in R, you can call this package to call some function directly for coding much easier. For somebody who don't know how to code, we provide a very online, very convenient, user-friendly Shiny app. You can just upload your data, click several buttons, and they will calculate for you. And I don't have time to show this, but I provide this link here. You can play with it. Sometimes for this online app, you need to upload your data to this external server to run our method. But sometimes your data maybe is from patient data. Patient data have very strict regulations. Maybe you cannot upload your patient data to outside of your institute or outside of your university. That's why we also provide a desktop software. You can just come to this My Lab website, download the software, and install on your local computer. In that case, your data would physically stay with your own computer. You will follow all the university policies. So that's the software proposed. As I mentioned, this paper was published last year in the 2022. It actually not only help Alzheimer's disease, we also help lots of other disease research. We also help the immunology. For example, this paper use this method that will help our collaborative published paper, immunology paper on Nature, and also on the Nature immunology on the molecular cell. That's a very helpful method. So after this, what's next? Think about the microscope proposed. So primitive confocal, you give a blue light, and then your protein will emit green fluorescence. That's called a green fluorescent protein. That's called confocal. But remember, this blue laser has a short wavelength.
Time check. Just we have 5 more minutes.
So this blue laser has a short wavelength, strong energy. It can damage your cell. But now we have some new technology called a 2-photon excitation microscope. It doesn't give you a blue laser, it gives you the laser with longer wavelengths and more mildly, more softer, like it give you 2 photons, like a red color, and then it still release a green color. By this, they can keep your cell alive and keep recording while your cell is active there. So this is a movie to show that. For example, this is from another similar disease, like a Parkinson's disease, very similar to Alzheimer disease. You can see some neuron cells have action potential, we call spike. Along the time, you can see this. By this new microscope, we can record this. This is a real data from Stanford University, from my friend's lab. So by this, for example, in the Parkinson's disease, you have control mouse and the Parkinson's disease mouse model. For the control mouse, you can see when you have the spike for these different neurons, they are randomly distributed along the time. But for the Parkinson's disease, many times they occur at the same time. When they occur at the same time, we think this is also called a co-localization. The co-localization is along the time. It's a one-dimensional polarization. So remember, our method can be used for the 2-dimensional polarization or 3D polarization. If you have 4D, you can also apply it. But it's a challenge to apply our method to the 1-dimensional polarization, like along the time. So how to do that? That's would be our challenge. That's our next target we want to address. So that's all my talk. Really appreciate the chance, opportunity provided from Dr. Des and also the NACC give me a chance to talk here to everybody here. And I always appreciate my employer, the Department of Preventive Medicine at the Northwestern Fiber School of Medicine, to support my research. This is my team, previous and current team members. What I present today mainly is performed by my previous postdoc, is Cheryl Liu. Now she's the assistant professor at the University of New Orleans. And another is the software supported by the Jiahui Xu. They were my biostatisticians. Okay, thank you. I'm happy to take any questions.
Yeah, thank you. That was great. So we all heard a range of talks, all the way from like de-identification of images to like PET images to like immunohistochemistry. So thank you again, Dr. Schwarz, Solingapuram, and Dr. Zhang. That was really great. And if there are any questions, if any of you have further questions, please feel free to post it on the mailing list and the Slack channel. We do have one for the ADRC data course. So I'd love to continue this conversation for these 3 amazing speakers. And so please, let's continue the conversation. And before I end, I wanted to do a shout out that the fall ADRC meeting is happening on October 18th to the 19th and 20th in San Diego. So please register for the Data Core workshop, which is on October 18th. So Hannah has helpfully put a link to the registration of the Data Core workshop. So please consider registering for the Data Core workshop. It's available both for attending virtually as well as in person. So I look forward to meeting many of you at the fall ADRC meeting and then perhaps AAIC next year. So, okay, so round of applause for our speakers. Thank you so much.